The topic for discussion today is multiple alleles and blood group inheritance along with the inheritance of RH antigen. You are aware that genetics is a modern branch of biology which deals with genes, their expressions and variations. The word genetics was proposed by William Bateson. You are aware that Mendel is recognized as the father of genetics, and T.H. Morgan, who worked on Drosophila, banana fruit fly, became popular in the world as the father of modern genetics. Mankind owes a lot to a great person who helped us to overcome incompatible blood transfusions, and nowadays blood transfusion has become very easy. That way we owe a lot to Landsteiner and his fellow workers who won the Nobel Prize for his epoch discovery of blood groups in the year 1930. Now, we are at times perplexed to see the gene magic. Now, this is one such interesting show about the role of gene. Usually, a gene has two alternatives or versions, which we call them alleles. రెండు ప్రత్యామ్నాయాలు ఉంటాయి ఒక జీన్కి. ఆ రెండు ప్రత్యామ్నాయాలను కలిపి మనం alleles, యుగ్మ వికల్పకాలు అని అంటాం. Now, these alleles are situated at the same locus of a pair of homologous chromosomes. సంగత క్రోమోజోముల మీద ఒకే లోకస్ వద్ద ఇవి ఉంటాయి. But many times at times a gene may have 3 or more alleles at the same locus of homologous chromosomes of an organism. So, when a gene has 3 or more alleles at the same locus of homologous chromosomes of an organism, they are referred to as multiple alleles. Now, what are these multiple alleles? Now, when 3 or more alleles of a gene exist in a population, the phenomenon is called multiple allelism. So, they can be studied only in a population, but you can't observe them in the genotype of a diploid individual. They are there in the population. So, a diploid individual possesses only 2 of those alleles that are there in the population. Multiple alleles always control the same character of an individual, and there is no crossing over among them, unlike the other alleles located on homologous chromosomes. Now, look at this great man whom we owe a lot. Landsteiner, an Austrian-born American immunologist, physician, pathologist, and he was awarded the Nobel Prize in 1930 for physiology or medicine for detecting the major blood groups and creating the ABO system of blood typing. He discovered these blood groups in 1901, but he became the Nobel laureate in 1930. Now, The discovery of blood groups revolutionized the processes of blood transfusions and medical practices related to the blood transfusions. Now, prior to the discovery of blood groups, we don't have the actual mechanism. Though blood transfusions were attempted prior to Landsteiner in several parts of Europe, sometimes they were found to be compatible, sometimes they were found to be incompatible. So, as more and more incompatible blood transfusions were appearing, they were banned. Now, after Landsteiner identified the ABO type of blood grouping in humans, blood transfusions have become easy and lives of millions of people have been saved all over the world. An excellent example of multiple alleles is human blood type AO. It is controlled by a triallelic gene located on autosome 9. Now, blood type is determined by the presence or absence of antigen on the surface of red blood cells, as well as the antibodies in the plasma or that serum fluid. Now, what is an antigen? Antigen is a substance, usually a protein in nature, that stimulates the formation of antibodies. Now, here the blood group antigens are not, say, simple proteins. In a way, they are the sugar polymers. That way, they are different from the normal antigens, which are mostly proteins. Now, they stimulate the formation of antibodies. Now, antigens are found on the surface of the red blood cells. They are bound to the membrane enclosing the RBC. Now, antibodies, because the reaction between antigen and antibody leads to incompatible blood transfusions. That is why we are to know about the antigens and antibodies in general. Antibodies are Y-shaped proteins formed within the blood, which react with the respective antigens. You learnt about blood group antigens which are present on the surface of the RBC. They are sugar polymers. Now, antibodies are Y-shaped proteins produced in the blood by what are called these B lymphocytes and their products plasma cells. They react with the antigens. Antibodies are found in the blood plasma or serum. Blood group antigens, mainly two, A and B. They are actually sugar polymers that are bound to lipid molecules on the surface of RBC. They are also called isoagglutinogens because they cause agglutination, 𝗬𝗏 of RBC in the case of incompatible blood transfusions. ఎప్పుడైనా అపసవ్య రక్త సంధానాలు జరుగుతూ ఉన్నప్పుడు ఈ రక్త కణాలు గుచ్ఛీకరణం చెందుతాయి, దాత రక్త కణాలు అన్నీ కూడా. ఎందుకంటే అదే రకము యాంటీబాడీతో ఎన్కౌంటర్ అవ్వడం వల్ల, దానివల్ల రక్త ప్రసరణకు అవరోధం కలుగుతుంది. దేర్ విల్ బి హిండ్రెన్స్ ఆర్ interruption for the free flow of blood. Blood flow will be blocked and the agglutinated RBC undergo hemolysis and releasing the hemoglobin also. Thus, the incompatible blood transfusions often lead to death of the patient. Now, ABO blood system is continued. The antibodies anti-A and anti-B are called isoagglutinins, which are usually IgM type. We have these antibodies IgM, IgG, IgE, like that. Each individual has a combination of elements from the two systems A, B and RH, which are used to type blood. Every individual has a combination of the elements, that is A, B, O, and the RH antigen, its presence are absent. So that way there are two systems, ABO system and RH system. So we have combination of elements of these two systems as phenotypes. The ABO system has 4 different phenotypes of blood, namely, what are they? A, B, O and AB. Now, the percentage of different groups across the globe varies from one country to another country, from one tribe to another tribe, from one region to another region, from one caste to another caste. But, you know, the person with the same blood group may be found in different communities, different countries, different religions. A unique thing. Now it shows universal brotherhood. At times, the blood group of a rival community, the person with the blood group of a rival community, with our own blood group from a rival community, will become our pranadata. Because even members of our own relatives may not be having our own blood group. So they are not suitable donors. The suitable donor is found to be from an opposite religion or opposite caste or a foreigner. So that way blood groups represent universal brotherhood. విశ్వమానవ సౌభ్రాతృత్వానికి రక్త వర్గాలు ఒక ప్రతీకగా పేర్కొనబడతాయి అని. So we know human beings, world population has been classified into these 4 blood types, A, B, O and AB. Now let us see how this classification is done. Now ABO blood system. Now blood group A, it accounts for 19.5% among Indians. So people of A group have A antigens on the surface of RBC, A antigens on the surface of RBCs and B antibodies in their plasma. You do not find the same antibodies. For example, a person with A blood group will not possess anti-A antibodies in his plasma. He will have anti-B antibodies in his plasma. Now blood group B. Now here B group people happen to be the predominant group of people among Indians in terms of blood groups. Now B group represents 40.5% of the total Indian population. Now people having this blood group B have B antigens on the surface of their RBCs and anti-A antibodies on their blood plasma. It is the most predominant group among Indians that you can see there also. Now ABO blood system, the next groups. Blood group O, next to B, we have more number of people with O group when compared to A and AB people. Blood group O represents 31.5% of the Indians. Now, people with the blood group O means they do not possess antigens, have neither A nor B antigens on the surface of their RBCs, but have both anti-A and anti-B antibodies in their blood plasma. So, they have anti-A, anti-B antibodies in their blood plasma. Why they possess the two antibodies? Because they don't have the corresponding antigens, antigen A and antigen B on their plasma, on their RBC. Now, the last group, but the least represented one also, blood group AB. Now, 8.5% it accounts for. Now, people of this group AB have both A and B antigens on the surface of the RBCs. Now, once a person possesses the two antigens on his or her RBCs, it goes without saying that the person doesn't possess the respective antibodies, anti-A antibody, anti-B antibody in his or her plasma. And as it is only 8.5% of our population, it is referred to as the least represented blood among Indians. That is why often we find scrolls in TVs, so and so, AB positive, AB negative. AB negative ante 0.5 or 0.4 ఉంటుంది. So, AB positive ante around this 8%, very rare. So, that is why they don't get the donor in time. The process of blood transfusion and life saving at times may become very difficult to perform. Now, we go into the genetical aspect and the inheritance of blood groups. The gene I has 3 alleles. I ante 𝗖𝗍𝗧𝗦𝗔𝗧 ani. Now, the gene I has 3 alleles, A, IA, IB and IO. Now, this gene is located on the first pair of homologous chromosomes, the alleles of this gene. A and B antigen molecules on RBCs are produced by 2 alleles A and B of the gene I. Now, the 2 alleles IA and IB are codominant. They are not dominant over each other. They are codominant. And both of them are dominant over IO allele. So, O is double recessive here. Now, allele A produces antigen A and allele B produces antigen B, and allele I, no doubt, produces a protein which cannot produce any enzyme, which is non-functional. Now, here you have the blood types A and B. Each has 2 genotypes, homozygous. Each has 2 genotypes, homozygous, IA, IB, IB. And heterozygotes are also there because A and B are dominant over O, AO and BO. But the 2 blood types A, B and O have each only one genotype, IA, IB, because they are codominant. O is double recessive, therefore it is II. Now look at this illustration, which is very much clear to you. Now the first one is this red blood cell type. Then you have the antibodies present and then antigens present. Now group A, the first figure, indicates where you have only A antigens on its RBC. And when you look at this plasma, A group person will have B antibodies. So antigens A. Then when you look at the group B, you find B antigens on the RBC and A antibodies in the plasma, and of course the B antigens. Then AB, both antigens you find on the RBC. There is a difference in the shape of those antigens too. When both antigens are present on the RBC, can you expect the respective antibodies to be present? If one antigen is present, the respective antibody will not be there, will be absent. Therefore, this AB persons, they have both antigens on the surface of their RBs, but the respective antibodies, anti-A and anti-B, are absent in their plasma or serum. Then you look at the O group. No antigens on the RBC. A antigen is absent, B antigen is absent, that is why they are not shown there. And then antibodies, you find both anti-A antibody, anti-B antibody. Both antibodies are present in O group. Now O group people have no antigens on their RBCs, but their plasma possesses anti-A antibodies, anti-B antibodies. So here antigens, no antigens, neither A nor B are absent. Now this is a beautiful illustration which will explain you, rather it is self-explanatory if you look into this and study it closely. Now here 2 copies of the gene I are inherited, one from the male parent and another from the female parent. Now the possible combinations of alleles that produce blood types along with antigens and antibodies compatibility of transfusions are given below. Now you have about 6 vertical columns in the slide and 5 horizontal rows. Now you, in the first place, you have the phenotypes of the blood groups. Now phenotypes, you have A, B, AB and O. Now for the A phenotype, how many genotypes are expected? Second column, genotypes. Now the genotypes are IA, it is a homozygote. Another one, IA, IO. That means a person with blood group A may be either a homozygote or a heterozygote. Next, here the antigen is A. Now antibody is B, anti-B antibody. Now to whom he can say, from whom he can receive the blood. He can receive blood from his own group A and the universal donor O group. Now to whom he can donate? A group people can donate their blood to which blood groups or which blood types of persons, that is A and AB. AB is universal recipient. So, A has A, his own group, and AB as recipients. A has two donors, his own group and O group. Then the B phenotype, just like A, B phenotype may be either homozygous or heterozygous. IA, IB, IB, and IB, IO or small I. Then antigen B. Antibody, not B antibody. Here anti-B antibody, it won't occur. Anti-A antibody you find here, because same antibody is absent. Then to whom B group person can donate his blood, that is to his own group B and O, from whom he can to whom he can donate his blood, that is he can donate his blood to AB and his own group. He can receive blood from his own group and O. Now AB, you find IA, IB, because they are co-dominant, both are present here. Now antigens present, A antigen is there, B antigen is there. When both antigens are present, there is no scope for the presence of antibodies. So both A, anti-A antibody, anti-B antibody are absent here. Now AB group people can receive blood. AB is branded as the universal recipient. Why is it called universal recipient? That we take up after this. For the time being, AB is a universal recipient. Therefore, AB group people can receive blood from any of the four groups, AB, A, B and O. But AB people can donate their blood only to the group of their own. They cannot donate their blood to any other group. If it is done so, it becomes incompatible, and that leads to the death of the recipient instead of recovery. Now coming to the O group. O phenotype has only one genotype because it is a double recessive, I. And then because A and B are dominant over O, therefore A, B groups have heterozygotes besides their homozygotes also. So 4 phenotypes and 6 genotypes you find here. Now O, antigens, both antigens are absent, neither A nor B is there. And then antibodies, when the antigens are absent on RBC, you find both antibodies, anti-A antibody is there, anti-B antibody is there, both of them are there in their plasma. Then to whom O can donate or receive his blood, from whom O can receive his blood. Now when O is the recipient, you find both antibodies. So they recognize any antigen A or B with A blood group or B blood group or AB. Therefore, for O blood group, A, B, AB as donors is banned. That is, they are banned. Now, this O, because it is without antigens, it can donate its blood to any person. Therefore, it is universal donor. That is, O has these, can be a donor to A, B, AB and O. Here is an illustration, you look at that. You have the O above, bottom AB, and on the right and left sides you find this A and B. Now O arrow is directed to A, B and AB. That means O can donate blood to A, B, AB besides its own group. Now AB, all arrows are directed, remaining 3 arrows are directed to AB. That means AB can receive blood not only from O but also from A and B. Now, persons with type O are universal donors because there are no antigens on their RBCs to trigger immune response. So, when there are no antigens there to be identified by the antibodies and interact with them, so it is miscible, compatible with all the remaining 3 types of blood groups besides O. Persons with AB type are universal recipients because they do not have any antibodies to recognize A and B antigens of the donor. Now, actually, in each blood group, when we take RH antigen into consideration, which is discussed later, if RH antigen is present, we say that is positive. If RH antigen is not there, we say negative. So, A positives are there, A negatives. B positives, B negatives. AB positives, AB negatives. O positives, O negatives. So, the most predominant group is O positive. Least represented group is AB negative. Now, who can act as universal donor? That is O negative, because RH antigen is also not there. So, this blood can be transfused to any person. It becomes compatible blood transfusion, irrespective of the blood group type of the recipient. Likewise, AB. AB is the universal recipient. AB, suppose negative, cannot be the recipient for the same blood type, that is positive blood type, for a second time. Because in the first time, the negative blood produces anti-RH antibodies having sensitized by the RH antigen present in the donor. Therefore, here if there is no RH antigen and in the recipient itself, if there is RH antigen also along with AB positive, AB, we say it is AB positive. AB positive means AB blood group people with RH antigen. Now, they are the universal recipients because they have the antigens, recipients. What is important here? The antibodies present in the recipient and the antigens on the RBC of the donor, these two should not be one and the same. Here, to identify and interact with the donor's antigens, there are no antibodies here. No A antibody, no B antibody, no this anti-RH antibody. Therefore, the true universal recipient is AB positive and the true universal donor is O negative, not O positive. Now, how to identify these blood groups? Now, blood typing, it is used to determine the blood groups of an individual. It involves the mixing of the blood with 3 different reagents, that is the reagent with A antibodies, reagent with B antibodies and RH antibodies. Now, here agglutination or clumping of RBC indicates that the blood has reacted with the antibody reagent and it becomes an incompatible blood transfusion if such a blood transfusion is attempted. For example, blood type A will clump with when it is placed in antibody A reagent. Blood type B will clump with when it is placed in antibody reagent B. Blood type AB will clump when placed in antibody A reagent and antibody B reagent, because it has both antigens. So it gives clumping with the reagents having anti-A antibody, anti-B antibody. Now, in the case of blood type O, O will not clump either in the reagent with A antibody or with the reagent B antibody. Why? Because there are no antigens to interact with these antibodies on the RBC of O type. Now, this is how the blood typing is done whenever the blood group is identified. It is a must for everybody to go in for this identification of his or her own blood group, because we do not know whenever we need this blood transfusion. If our blood typing is there with us, it will be easy for us to go in for the donor's blood type and have quick blood transfusion so that recovery will be speeded up. Now, importance of the blood types in blood transfusions. Ante compatible blood transfusion, incompatible blood transfusion, and in solving medico-legal cases also, blood groups help us a lot. While transfusing blood, the type or types of antigens of the donor and the type or types of the antibodies present in the plasma of recipient, they are to be taken into consideration. And how about the remaining? The antibodies of the donor and the antigens of the recipient are relatively of less importance. We don't give them any weightage because you don't find much interaction with them. So what is important here is the antigen present on the RBC of the donor blood type and the antibody present in the plasma of the recipient. Say A blood group is incompatible for B person or vice versa. A is incompatible for O group, but O can donate his blood to A because A is recipient. O, though A has anti-B antibody and O has no antigen to interact with that. So, that way this compatible and incompatible blood transfusions, one should have the knowledge of these blood groups. Now, other thing is blood groups help us in solving medico-legal problems. How is it so? A couple, wife and husband are there. They claim the O group baby as their child. But when the blood groups are analyzed, the blood group of husband is homozygous A. Blood group of wife is homozygous B. There is no scope for this couple to get a O group child. Therefore, they are proxy people. They are claiming the child of another parents as theirs. So, it should not be like that. So, that way we can predict the blood groups, probable blood groups of the children if we know the genotypes of the parents in question. This is the other important benefit that we get from the knowledge of these blood groups. So far, we have discussed about the ABO type of blood system. Now, the RH type of blood system. RH antigen. The name RH antigen refers to antigen that was first discovered in the blood of rhesus monkey. As such, it has earned that name RH antigen or RH factor. If RH person, now the RH factor was discovered by Landsteiner and Wiener. RH antigen or RH factor so named because it was first identified on the surface of RBC of rhesus monkey and later on, of course, observed in human beings also. The term RH factor refers to the D antigen, which is the dominant one, D antigen of the RH system. Now, a person can be either RH positive or negative depending on presence or absence of RH antigen, that is antigen D. Most people fortunately are RH positives, especially among Indians in the 4 blood phenotypes A, B, O, AB. Less than 5% negatives. The remaining 95-96% are positives, which is slightly different among Europeans and Americans. Among the Europeans and Americans, the white race, they have nearly 15% of their population as RH negatives. Only 85% are RH positives. Thus, some 20 years ago, marriage between a RH positive male and RH negative female was not at all favored. Why? The child, the second conception onwards, the baby gets aborted before birth. So that way, they used to be careful in selecting their mates. It is, if the male is negative, no problem. If the male is positive and the female is negative, then the problem comes. And we will see after some time how the problem comes. RH positive or RH negative blood groups depends upon presence or absence of antigen D. Most people, luckily or fortunately, are RH positives. A anti and D antibodies are absent in the plasma. Anti-D antibodies are absent in the plasma. Now, if RH negative person is exposed to RH positive blood cells for the first time, anti-D antibodies are formed in the person's plasma because the negative blood gets sensitized for the contact with antigen D in the form of RH positive blood cells. And if you want to give a consecutive second blood transfusion to the same person who has received RH positive blood and the recipient is negative, even if you transfuse the same group of positive blood, it becomes incompatible blood transfusion. RBC of the donor get agglutinated because the antibodies the anti-RH antibodies or anti-D antibodies that are there already formed, they bring out agglutination of the donor's blood cells. Therefore, it becomes an incompatible blood transfusion. But on the other hand, Rh negative blood can be transfused to Rh positive person without any consecutive effects or complications. Why here the Rh negative blood, no Rh antigen, and recipient is already having the Rh antigen. Therefore, he doesn't have any Rh antibodies. So, no Rh antibody to interact with this. So, it becomes compatible blood transfusion. Now, here we say which marriages became biologically incompatible. And if both are Rh positives, male and female, no problem. If both are Rh negatives, male and female, no problem. If the female is Rh positive, male is Rh negative, no problem. The problem comes with Rh positive male having Rh negative lady as his wife. Rh positive is dominant over negative. So, when she conceives, she is likely to conceive a positive baby only because the Rh positive or her hubby is dominant over that. So, when she conceives a Rh positive baby in the first conception, during the course of pregnancy, the fetal RBC having Rh positive antigen, D antigen, may escape into the maternal blood through placental circulation. Thus, the maternal blood gets sensitized and produces anti-D antibodies. So, in the second consecutive or successive pregnancy, when she conceives the Rh positive baby, these anti-Rh antibodies or anti-D antibodies that are there in the plasma of the mother find their way through this placental circulation into the fetus and attack the fetal RBC, thereby causing agglutination of fetal RBC and hemolysis even before birth. Therefore, it becomes erythroblastosis fetalis or hemolysis of newborn, HNB. So, that is an important point one has to remember here. The Rh factor involves 3 different pairs of alleles located on 3 different closely related loci on first chromosome pair. This is according to Fisher and Race. Now, all genotypes with a dominant allele D will form Rh positives. Now, Wiener hypothesis, the second one. Now, here instead of 3 pairs, here there are 8 pairs at a single Rh locus. Now, all genotypes with capital R, that is the dominant allele, form Rh positives, and the double recessive small r, they form Rh negatives. So, according to this Fisher and Race, 8 pairs. According to this Wiener, only 3. According to Fisher and Race, 3 pairs, and here according to Wiener, 8 pairs of alleles. Now, it can be a problem, as I told you just now, if a Rh woman is pregnant and the fetus is Rh positive, because when her hubby is Rh positive is dominant over Rh negative. So, that way, if he is Rh positive homozygous, then every child of this couple will be Rh positive. And during the course of pregnancy, there will be certain times when the baby's blood may enter the maternal blood through placental circulation. Look at this. You can see the womb and the placenta. If the mother is Rh negative and is exposed to her baby's Rh positive blood, the mother's blood produces antibodies ante anti-Rh antibodies or anti-D antibodies against the baby's red blood cells. Now look at this. Normally a problem. The problem is only when an Rh negative mother conceives Rh positive baby. So here this in the course of the first pregnancy, on exposure to Rh positive antigen, when the fetal blood cells escapes into the mother, the maternal blood gets sensitized and produces anti-Rh antibodies. But the buildup of these anti-Rh antibodies will not be sufficient to cause damage to the fetal red blood cells. Therefore, the first delivery is a normal one. The problem comes with the subsequent deliveries. Suppose if in the next delivery also, this mother whose blood has this anti-Rh antibodies or anti-D antibodies already produced during the course of first pregnancy, conceives a second consecutive Rh positive baby, the antibodies find their way through placental circulation into the fetus, thereby causing agglutination and hemolysis of the fetal RBC. Therefore, the fetus undergoes erythroblastosis fetalis, or what is called this hemolytic newborn disease. So, this problem nowadays has been solved. If the problem is identified, this Rh negative mother is given certain injections which will inactivate the Rh antigen antibodies that are formed just before delivery of the first baby or a few days after parturition. So nowadays this is not a major problem. So this is about Rh antigen. Rh antigen knowledge is essential for effecting successful blood transfusions and also for getting normal deliveries in case the mother is Rh negative and the father is Rh positive. You know, there is a formula to find out the kinds of numbers of genotypes. The formula is half into n into n plus 1, where n is the number of alleles. Say for example, triallelic, our blood groups, 3 alleles, IA, IB, IO. So what is the number of alleles here? 3. So half into 3 into 3 plus 1, that is 4. 4 into 3, 12. 12 by 2, that is 6. So you have 6 genotypes here. That way you can arrive at tetraallelic 10 genotypes, and this octoallelic you can arrive at 36 genotypes using the same formula, half into n plus 1 formula. Now, this is helpful in finding out the kinds of number of genotypes the multiple alleles in question can generate or produce. So, have a nice day, and this is a very important topic even from the normal life point of view also. I hope you have all enjoyed this lesson. Thank you very much.